Welcome to Chem 332. This is the first lecture. What we will start out with is Chapter 17, which is an introduction to metabolism. And I'll just correct the date here. The date is going to be, this is the lecture for Wednesday, January 22nd. So this chapter is going to address metabolism. If you took Chem 331, you focused on the macromolecules of biochemistry. You learned about thermodynamics, you learned about proteins, nucleic acids, carbohydrates, lipids, the specificity and catalytic power of enzymes, the regulation of enzymatic catalytic activity, and the transport of ions and molecules across membranes. And so now what we're going to do is we're going to use this foundation of biomolecules to answer the following questions. So we're going to look at metabolic maps. We're going to look at how anabolic and catabolic processes form the core of metabolic pathways, and I'll explain those terms later on in the lecture. We're going to determine what the energy currency of the cell is, how a cell extracts energy in reducing power from its environment, and then we're going to look at the experiments that can be used to elucidate metabolic pathways. For now, we are not going to look at the last question. So here is a metabolic map. If you look at this map, it obviously doesn't look like very many road maps that you've encountered, or maybe it does. It's a complex series of chemical reactions, and it's an integrated network. Now, collectively, this is known as metabolism or intermediary metabolism. And there are hundreds of enzymatic reactions organized into discrete pathways. And these pathways proceed in a stepwise fashion, transforming substrates into end products through many chemical intermediates. Now, even though there are more than 1,000 chemical reactions in even as simple an organism as E. coli, there is a coherent design to metabolism, and there are many common motifs. And these motifs include the use of an energy currency, which is going to be ATP. And we'll look at ATP in more detail later. And the repeated appearance of a limited number of activated intermediates. And so just to simplify things a little bit more for you, okay, these are the metabolic pathways that we are going to explore this semester. We're going to look at carbohydrate chemistry, we're going to look at the TCA cycle, we're going to look at how fats are synthesized and broken down, and how nitrogen is metabolized. To make it even simpler, we're going to look at Biochemistry Land. So first we're going to start in Carbohydrate Land. And here we're going to look at glycolysis, gluconeogenesis, and glycogen metabolism. From there, we're going to move on to the main powerhouse of Biochemistry Land. The main powerhouse includes the tricarboxylic acid cycle, electron transport, and oxidative phosphorylation. Then we will explore lipid land, in lipid land we are going to look at fatty acid synthesis and breakdown. We will also get, and I'm going to write this in red so it comes across here, to the amino acid. And this is where we'll look at nitrogen metabolism. Here, after we cover amino acids, we will look at metabolic integration, biosignaling, and metabolomics. Now, while we are in biochemistry land, we will also visit the infirmary, and this is how we will look to see how errors in metabolism affect health. So the purpose of this chapter, and I'm going to go a little bit beyond of what's in the chapter, is to introduce some general principles and motifs of metabolism. Okay? So there are going to be 5 principles that we're going to explore. The first one has to do with the fact that fuels are degraded and large molecules are constructed step by step in a series of linked reactions. The second principle is that there's an energy currency common to all forms of life, which is ATP. And ATP, or adenosine, triphosphate, it links energy releasing pathways with energy requiring pathways. The third principle is that the oxidation of carbon fuels powers the formation of ATP. The fourth principle is that although there are many metabolic pathways, there are a limited number of reaction types and there are intermediates common to many pathways. So if you think back to that map that I showed you in the beginning, in I think it was the third slide, you could see that the dots, they represent intermediates, and the lines represent the pathways. So that there are dots that are common to many of the pathways that they're shared. The fifth principle is that metabolic pathways or metabolism is highly regulated. Alright. So we're going to keep these principles in mind as we study metabolism this semester. Now, the whole purpose of metabolism is energy. Okay, so that's the common currency, and it's the common theme. And why do we need a continual input of energy? Well, there are three purposes. We need energy for mechanical work. This is your muscle contraction and cellular movements. We need energy for active transport. And this includes transport of molecules and ions. And we need energy for the synthesis of macromolecules. So where does all this energy come from? Well, the free energy used in these processes to maintain an organism in a state that's far from equilibrium is derived from our environment. And we can classify organisms based upon their different carbon requirements. Now, autotrophs, okay, so that includes photosynthetic bacteria, green algae, and vascular plants. They use carbon dioxide as their fuel source, and they construct all their carbon-containing biomolecules from carbon dioxide. Some autotrophs can also use atmospheric nitrogen to generate all their nitrogenous biomolecules as well. Heterotrophs, on the other hand, cannot use atmospheric carbon dioxide. We obtain carbon in the form of complex molecules such as glucose. Most multicellular organisms and microorganisms are heterotrophic. In our biosphere, autotrophs and heterotrophs live together in a vast interdependent cycle in which autotrophic organisms use atmospheric carbon dioxide to build their organic building blocks, and some of them generate oxygen in the process. Heterotrophs, in turn, use organic products that the autotrophs produce. So we use glucose, okay, and we return carbon dioxide to the atmosphere. Some of the oxidation reactions that produce carbon dioxide also consume oxygen, converting it to water. Thus, carbon, oxygen, and water are constantly cycled between heterotrophic and autotrophic worlds, with solar energy as the driving force for these global processes. In contrast to the cycling of carbon, oxygen, and water, energy is constantly transformed. So when we looked at that big metabolic map, we can see that metabolism consists of energy-yielding and energy-requiring reactions. And so we can break it down into catabolism and anabolism. Metabolism is essentially a linked series of chemical reactions that begins with a particular molecule and converts it into some other molecule or molecules in a carefully defined fashion. In metabolic processes and in all energy transformations, there is a loss of useful energy, which is the free energy, which is delta G, and an inevitable increase in the amount of usable energy, heat and entropy. So this is free energy, and if you remember, delta S equals entropy, and delta H is your heat. We can divide metabolic pathways into two broad classes. There's those that convert energy from fuels into biologically useful forms, and those that require inputs of energy to proceed. Now, reactions that transform fuels into cellular energy are called catabolic reactions. So on the left side, we have catabolism. Catabolism is oxidative, so that we oxidize carbohydrates, fats, and proteins. And in that oxidation, we produce ATP and energy-poor products such as water, carbon dioxide, and ammonia. Reactions that require energy, such as the synthesis of glucose, fats, and DNA, are called anabolic reactions, or anabolism. So we have anabolism here on the right side. And this is where we use simple precursors, such as amino acids, sugars, and fatty acids. And we have a reductive process that results in the synthesis of proteins, polysaccharides, lipids, and nucleic acids. And in this process, ATP is consumed. Now this figure was taken from your book, and really what you need to take home from this figure, it's looking at metabolic regulation. Now anabolism and catabolism can occur simultaneously in the cell, but a cell needs to have a mechanism or a way of regulating the processes. Now, if catabolic pathways were the inverse of anabolic pathways, then at equilibrium, nothing would get accomplished. So there have to be some differences in anabolic and catabolic processes. And typically those differences are going to occur at regulatory points. Okay. In addition, a regulatory point is typically thermodynamically favorable. Meaning it's not reversible. The other way in which a cell will regulate metabolism is by compartmentalization. So that Anabolic processes will occur in one compartment of a cell, and catabolic processes will occur in another. Right, now we come to ATP, which is the universal energy currency. And we'll go into a little bit more detail in this section than your book does. Cellular metabolism is facilitated by the use of this common energy currency. Now, most catabolic reactions consist of reactions that extract energy from fuels such as carbohydrates and fat, and they convert this energy into ATP. When you hydrolyze ATP, it's exergonic, or exothermic, and this hydrolysis is what drives your anabolic reactions. Now let's look at the structure of ATP, and it will help us explain why this is the case. First, let me just go through, again, this figure on the right. Okay, so if we look at the blue line, when we have ADP and we make ATP, it's from the oxidation of few molecules. So that's where you're oxidizing your carbon sources. When we hydrolyze ATP, this is what drives motion, active transport, and your anabolic biosynthetic processes. So if we look at the structure of ATP, we have an adenine, we have a ribose, which is a 5-carbon sugar, and we have this triphosphate. Now all the energy comes from the triphosphates. So it's going to be a phosphoryl group transfer. That's where the energy lies. So let's look at the structure of ATP and see why it is the energy carrier. If we look at ATP, we can see that we have an anhydride bond there and an anhydride bond there. Okay. When we hydrolyze ATP, what happens is we get greater resonance stabilization. So what happens is that when let me just label these. This first phosphate is alpha, the second is beta, the last is gamma. If we were to hydrolyze the gamma phosphate, we would get inorganic phosphate, depicted as Pi, now we could delocalize this negative charge to the oxygens. In addition, there's a fourth resonance form where there's a positive charge on the oxygen and all the other oxygens are negative. So we have greater resonance stabilization because we have these 4 new resonance forms, and then we can still have resonance on the oxygens of the beta phosphate group. In addition, ATP is readily hydrolyzable because of electrostatic repulsion. If we look at the structure of ATP, if we look at the structure of ATP, at pH 7, there are 4 negative charges. This results in repulsion. When you hydrolyze, you relieve or you reduce the repulsion. And finally, I'm going to squeeze this in up top here. The third reason that ATP is readily hydrolyzable is because of hydration. Inorganic phosphate and ADP is readily hydrated compared to ATP. Now let's look at some of the thermodynamics of ATP hydrolysis. When you hydrolyze ATP plus water, you get inorganic phosphate and ADP. Now, the delta G standard for this reaction would be negative 30.5 kilojoules per mole. Physiologically, the number is actually around negative 50.2 kilojoules per mole. And this can vary based on the ions present. So if I didn't mention this before, ATP is often associated with magnesium 2+ or manganese 2+. Now, this very negative free energy is used to drive reactions that require an input of energy. So this hydrolysis is used to drive thermodynamically unfavorable reactions. Some of those reactions are muscle contraction, protein conformational changes. You can have active transport of ions, and the synthesis of biomolecules. I just want to show you an example of how ATP can be used to drive an unfavorable reaction. So let's say we have A and we want to convert it into B. Now the delta G for this reaction under standard conditions is 16.7 kilojoules per mole. So it is an unfavorable reaction. If we were to calculate the equilibrium constant for this reaction, if you remember, for equilibrium constants, it's products over reactants. So it would be the concentration of B at equilibrium divided by the concentration of A at equilibrium. If we use the delta G to determine this, we would find that KEQ would equal 1.15 times 10 to the minus 3. What this means is that the net conversion of A into B cannot take place when the molar ratio of B to A is equal to or greater than 1.15 times 10 to the minus 3. A can be converted to B under these conditions if we couple the reaction 2 ATP hydrolysis. So, when we couple the reactions, the net equation looks something like this. Now, when we combine reactions, we can sum the delta G's, and we are going to multiply the equilibrium constants. Overall now, the new delta G under standard conditions would be negative 13.8 kilojoules per mole. And we get that from negative 30.5 plus the 16.7 kilojoules per mole. Now we have to multiply the equilibrium constants. So, if we multiply the equilibrium constants, it would be the concentration of B at equilibrium over the concentration of A at equilibrium times the concentration of ADP at equilibrium, inorganic phosphate at equilibrium, divided by the ATP concentration at equilibrium. If we do this in terms of delta G's, we would find that the new equilibrium constant is 2.67 times 10 to the 2. Now, if we compare these, we would see that now the ratio of now the ratio of A to B is 10 to the 5 orders of magnitude larger. Okay. In other words, by coupling the reaction to ATP hydrolysis, we can shift the equilibrium ratio by a very large factor. Now, if we were to look at ATP hydrolysis under physiological conditions, the delta G is negative 50.2. So we could even shift the equilibrium ratio even further. So keep this in mind as we go through metabolism, that ATP hydrolysis can be used to thermodynamically drive an unfavorable reaction. So this slide here is just summarizing what we discussed, why ATP serves as the energy currency. The first part here is showing you the hydrolysis, where you have water attacking the gamma phosphate, and then you break this phosphoester bond. A driving force for this reaction is resonance stabilization, relief of charge repulsion, and better hydration. And this here is showing you that resonance stabilization that I drew earlier. Now this table here looks at phosphoryl group transfer potential. So that the idea is that when we lose this Pi, we're transferring it. Now if you look, you can see that ATP is in the middle, not in the middle, but it's intermediate. In the table. And the reason why ATP is intermediate is because it will enable ATP to function efficiently as a carrier of phosphoryl groups. So there are times when we need to make ATP. So for example, in glycolysis, which we'll talk about on Friday, phosphoenolpyruvate, or PEP, will transfer inorganic phosphate to ADP to make ATP because it's higher up in the table. It has a larger free energy when you hydrolyze that phosphoryl group. We also can see phosphocreatine here. Now, if we think about this in terms of an athlete, the amount of ATP in muscle suffices to sustain contractile activity for less than a second. Creatine phosphate stored in vertebrate muscle serves as a reservoir of high potential phosphoryl groups that can be readily transferred to ADP. So indeed, when we're exercising, creatine phosphate is used to regenerate ATP from ADP, and the reaction is catalyzed by creatine kinase. This is why athletes often take creatine as a supplement. Now, once the supply of creatine phosphate is depleted, ATP has to be generated through metabolism. And you're going to generate ATP, as I've stated earlier, through the oxidation of carbon fuels. So we're looking at the oxidation of glucose and fatty acids. Now, if you remember back to organic chemistry, oxidation is the loss of electrons. It is also a gain of oxygen. So here we have a fully reduced carbon, and in carbon dioxide we have a fully oxidized carbon. And there's a spectrum, and carbon dioxide is the ultimate product. Of oxidation of carbon fuels. Now, the more reduced a carbon, the more energy released. Let's look at the structure of glucose and the structure of a fatty acid. So if you look at glucose, which is a 6-carbon sugar, so here's glucose. Here's a fatty acid chain. Now, if you look at these two molecules, glucose has a greater oxygen content. The carbons are more oxidized than the carbons in a fatty acid. If you look in the fatty acid, you mostly have CH2s. This is why you release more energy when you burn fats. Or when you use fats as fuel. However, the body does prefer to use glucose. So I've talked a lot about common intermediates in metabolism. And now we're going to look at those common intermediates. So we looked at ATP, and that's the energy currency. So let's look at the common metabolites that are present. So one of the common metabolites that you'll see is NAD+ and NADH. Now, when we talk about oxidation of our carbon fuels, we have a loss of electrons. Where do these electrons go? Well, the ultimate electron acceptor in oxidation is oxygen, O2. However, as we go along a metabolic pathway, electrons are not transferred directly to O2. Instead, our fuel molecules are going to transfer electrons to special carriers. These carriers are going to have either pyridine nucleotides or flavins. And then the reduced form of these carriers then transfer their high potential electrons to oxygen. So the first one that we're going to look at is nicotinamide adenine dinucleotide. So here again is the adenine. We have the ribose sugar. Okay. And here is your nicotinamide ring. This is your amide bond. Now, the reactive site is this carbon here. So we're either going to gain a hydrogen that has 2 electrons, and then that's how we convert NAD+ to NADH. Notice too that in the oxidized form, the nitrogen bears a positive charge. This charge is lost in the reduced form because you break the aromaticity of the ring. This slide here is just showing a blow-up of the nicotinamide adenine dinucleotide, and you can see again, this is the reactive site. This is where we are going to gain 2 electrons in the hydride anion. And then another type of electron carrier that we'll see is FAD, which stands for flavin adenine dinucleotide. So again, we have our adenine and our ribose. This portion here, which is the reactive site, is the flavin molecule. We have 2 nitrogens. Now, similar to NAD+, we're going to accept 2 electrons. But instead of accepting them in the form of a hydride anion, FAD will accept them as 2 protons accompanied by 2 electrons. So that we're going to protonate the 2 nitrogens. I do need to add a correction. It's FAD. This is the oxidized form. And it gets converted to FADH2, which is the reduced form. And you can go backwards with this as well. So the FAD is uncharged. So the first 2 carriers I showed you, the NAD+ and the FAD, they accept electrons and are used to transfer their electrons to oxygen in an oxidative process. In order for anabolic reactions to occur, we need a high potential electron donor. And so we need a donor because the precursors are more oxidized than the products. So anabolic processes are reductive, and the reducing power is needed in addition to the hydrolysis of ATP for biosynthesis. So if you look on top, for example, in order to synthesize a fatty acid, we have a ketone group that's oxidized in a 2-carbon unit here. And we need to make it into these methylene groups. And so we are going to reduce the carbons. So in addition to ATP, we need an electron donor instead of an electron acceptor. And that electron donor is going to be NADPH. Now, you can see it's very similar to the NADH. Here, at this position, the R is a phosphate group for NADPH. NADPH is used in an anabolic reductive process. It is a donor. NAD plus is used in an oxidative process, and it is an acceptor of electrons. Now, the phosphoryl group is important on this 2' hydroxyl of adenosine because it enables enzymes to distinguish between a high potential electron donor as opposed to a high potential electron acceptor. So it serves as a tag. Continuing on with our common carriers or common intermediates, we have acetyl-CoA. And so acetyl-CoA is abbreviated here on the bottom. Now the CoA can be represented up here. We have an adenine and our ribose. We have this pantothenate unit and a beta-mercapital ethylamine unit. Now, acyl groups are important constituents both in catabolism, in the oxidation of fatty acids, and anabolism, because they're used in the synthesis of membrane lipids. Acyl groups are linked to coenzyme A by a thioester bond. This is your thioester bond. Just to refresh your memory, this would be a regular ester bond here between the carbonyl and the oxygen. Now the reason why a thioester is used is because hydrolysis of a thioester is thermodynamically more favorable. The reason is that you have less resonance when you have a thioester than when you have an ester, than when you have an oxygen ester. So if I come up here, in an ester you can delocalize the lone pair. And have a plus charge on this oxygen. This interaction is weaker and not as stable in the thioester. So that is why a thioester is used for this transfer of the acetyl groups. Now, the use of activated carriers illustrates two key aspects of metabolism. First, NADH NADPH and FADH2 all react slowly with oxygen in the absence of a catalyst. Likewise, ATP and acetyl-CoA are hydrolyzed slowly without a catalyst. Because these molecules are kinetically stable, what this allows for is that catalysts, typically in the forms of enzymes, exert exquisite control because they are essential for these reactions. They control the flow of free energy and reducing power. The other thing that's important to recognize is that most interchanges of activated groups are carried out by this small set of carriers. Almost all the activated carriers that act as coenzymes are derived from vitamins. Vitamins are organic molecules that are needed in small amounts in the diets of some higher animals. Prior to serving as a coenzyme, you have to modify vitamins. So we can see here vitamin B5 pantothenate. This forms part of coenzyme A. Okay. Here we have niacin, which is part of the NAD plus and NADP plus. Here you can see where flavin comes from in vitamin B2 of riboflavin. We will see that they bring unique chemical functionality to certain enzyme reactions. So we can break down the complicated metabolic map into 6 types of reactions. We have oxidation-reduction reactions. Useful energy is often derived from the oxidation of carbon compounds. Typically, you have NADH and FADH2, which carry electrons to oxygen, the final electron acceptor. We have ligation reactions. So here we have bonds that are formed by using free energy from ATP cleavage. We have isomerization reactions where intermediates will rearrange where atoms in a particular molecule will rearrange and form a new intermediate. We have group transfer reactions where we transfer functional groups. This can be in the form of a phosphate group or an acetyl group. We have hydrolytic reactions, again where we're cleaving bonds by the addition of water. These hydrolytic reactions are seen when proteins are digested. And we can also have the addition of functional groups so that they can be added to double bonds to form single bonds or removed from single bonds to form double bonds. The enzymes that catalyze these types of reactions are called lyases. For metabolic regulation, There are three main modes of metabolic regulation. We can control the amount of enzyme, and this comes from transcriptional control and translation. So this is on the genetic level. And this can often be turned on and turned off by intermediates. We have catalytic activity. Okay, this is typically your feedback inhibition. And allosteric regulation. Where you can change the affinity of an enzyme in the presence of intermediates. Hormones can also be used to regulate catalytic activity. In addition, the energy charge of the cell regulates catalytic activity. So here you're looking at the ratio of ADP to ATP. And so depending upon the energy charge of the cell, an enzyme may be activated or inactivated. If it's a catabolic enzyme, it will be inhibited by high ATP. If it's anabolic, it's stimulated by high ATP. The third way in which we regulate metabolism is by controlling the accessibility of substrates. Now this can be done by compartmentalization, this will segregate reactions. This can also be done by different types of biosignaling, where you have messengers that activate pathways. So we've talked about some general trends in metabolic pathways, the common carriers that we're going to encounter, the energy currency of a cell. Now we're going to talk about some methods to reveal metabolic pathways. So your book talks about the initial experiments that were done with enzyme inhibitors. And so this is where an inhibitor would be added. Intermediates would build up. And then you could infer its role in the pathway. Okay. So now, as genetic methods have evolved, we can do mutations and knockout and knock-in experiments where we control the enzymes that are transcribed and translated. And by doing so, again, we can identify points in a pathway based on the intermediates that are formed or not formed. We can use isotopic tracers. So here we are using radioactive isotopes. Such as carbon-14 and oxygen-18. And you're using these as tags. And what you can do is you can track the flow of carbons. And then you can determine their fates. And you can either look at things by mass spec, or you can quantify the amount of radiation on a blot. NMR, which is a tool that you encountered in organic, can be used. It's non-invasive, and a lot of NMR is based upon tracing P31, which is a naturally occurring isotope of phosphorus. So here what this example from your book is showing you is that this is the phosphate that's attached to phosphocreatine, and you can see that it's in high concentration before exercise, and as we discussed, as we need phosphocreatine to make ATP, you can see that the amount of phosphocreatine decreases during exercise because the PI is being used to make ATP, and you can see an increase in that inorganic phosphate peak. So this concludes what you need to know from Chapter 17. I will post homework questions, and in Wednesday's class, we're going to use this information to analyze a case study, and I'll also take any questions that you might have pertaining to the lecture.